Good evening, good afternoon, and good morning. This is the 2089th day of continuous webinar conducted by International Forum for Promoting Homeopathy. Today, our guest is Dr. MASHHOOD. Let us begin the session with 1 minute silent prayer. Thank you all. The International Forum for Promoting Homeopathy comprises of homeopathic lovers, students, and doctors, and we have 3 sessions every day. As you all know, the first session of the day is already over in Hindi from 7:30 to 8:30. From 8:30 to 9:30, we have the international session in English, and from 9:30 to 10:30, we have a local language session in Malayalam. We are uploading the videos of the same on the very next day itself so that those who miss the session can watch it again. You can subscribe the same without paying any money. You can share it with anyone who loves homeopathy. The purpose of this forum is to take homeopathy to every home around the world. And let me come to today's guest. And today our guest is Dr. MASHHOOD. The doctor has completed BHMS and diploma in acupuncture. He is a retired medical officer from ICE Department, Kerala, and is doing a lot of research work in homeopathy, and particularly in plants and microscopic study of changes in homeopathic medicine. He is an author of 2 books, and the Unveiled Microsecret of Homeo and Herbals, and the second one is The Elements of Human Body. He has more than 37 years of clinical experience and is a wonderful personality. Has presented so many sessions in English as well as in Malayalam sessions. As you today, Doctor will be talking about homeopathy for plants, principles of action. That is Modus Operandi as well. This is a very, very important subject, as you know. The homeopathy, how it works on plants, and nobody knows. How we can apply the homeopathic medicine to these plants. It's entirely different, and so let us see how the homeopathic medicine works in plants. And so we proudly welcome the guest of the day, Dr. MASHHOOD. Welcome, Doctor.
Respected all Ifph members, sir, I am audible here.
Yes.
Okay. Doctors, coordinators, our beloved Dr. Samuel Hanneman, beloved my teachers and all others, my thanks all for this opportunity. Today's my topic is, I'm going to share this.
Yes.
Yes. See, I think that it can be seen.
Yes, it's visible.
Yes.
Visible.
Today's my topic is homeopathy for plants, acting principles. It is the modus operandi. So many factors are here. Medicines, then plants, then how these medicinal homeopathic medicines acts on plants, whether it is acting, whether there is any action on plants. These were the intention for my studies for these many years. Plant cells function with the chemical nutrients and their interactions. This is what I want to say. For these so many months, I am taking these class continuously about the action of medicines in plants and their reactions. Today, the most of the pictures I am included in this class is to see what results when homeopathic medicines applied to plants and keenly separated or observed experimental with the experimental mind. These plants shows so many changes with the naked eye we can see. But my study ends, or not ends, but it reaches to this point that I am now so much interested to say you about the microscopic study of elements which are taking place inside the plants after using these our dynamic potencies. If all of us know there are so many micronutrients, so there is a line one we have to keep inside, kept aside by these studies. These are not acting as micronutrients. First of all, I want to say that is the result. That is what is one I have seen. Secondly, our higher dilutions, even micronutrient level, they are producing very high changes in their elemental structure. That is what I confessed that plant cells function with the chemical nutrients and their interactions. As in our own body, also, what is happening is this in disease states. We can go to the slides. See, these are the very minute microscopic. See the leaf structure, that the cells you can see. These are minute microscopic plant insects, very small. I found in some peperomia plants which I experimented. And for this, why? Because presence of even the Silesia medicine, our homeopathic one potency, these insects were so far, it's not observed anywhere within that locality. After using Silesia, that is was the important one. That's why I first introduced this. This will in later older classes also I told you. Then crystals. This is the exact study of what happening, what is doing by me, crystals observations. There are crystals which are separated from the study of these solutions made from the leaves or the roots or the covering of the fruits, which are made into solution as our homeopathic preparation, and then they are under a microscope studied after several days. So these are the observations which I introduced here. The crystals now today there is no time to say all. In former classes you can see in YouTube, and I think that they are all helping for this class. The crystals are natural and induced, two types. That is this is my categorization. This is my own categorization. That is naturally we can see so many crystals when under microscope from a leaf or a plant, any part, root, everything. There are differs also. But what is the difference between the induced one and natural one? Induced means after using a fertilizer, after using any separate nutritional point or some other factors like homeopathy or any other medicines, dynamized, potent-based medicines especially. Then what we see is induced medicines. There may be some crystals which are normal as natural. We can see some natural crystals. Sometimes normal but not natural. Normal but not natural, their size may be increased, their shape may be deviated. Abundance, their presence, abundances may be more or less. These are all to be watched. For many experiments, we can have to assess what are these changes. It is not an elaborate study. Now it is not an elaborate study. For these years, first I was confused. Later I was calculated and characterized like these. Now it is very clear. Then the abnormal crystals may be there. There are some abnormal crystals after medicines, after some fertilizers, or after some successed medicine, after unsuccessed medicine. There are all abnormalities of the normal crystals. First we see some natural or normal crystals from any plant. So these plants which I collected, I observed were about most of the plants are chili. That is the chili plants, or other is the peperomia, means a small plant which I will show. So the abnormal crystals also. Then there may be some new type of crystals appears. The new type of crystals. So normal crystals may be there, normal but not natural. Then abnormal crystals may be deposited. Then new crystals also may be seen from these studies. See, for this, it is a plant. I am going very speedily because so many pictures are there. Only the time is on our 14 minutes. So I think that it is better to say some pictures about and have to assess what it is. You can see this is a plant which I used several medicines. This from this is after 6 or 5 months after there was no change. This plant was then February 14, 25. This plant is picture is taken. This now it shows so many flowers and a very good growth and food. There it is a course of the medicines which I used. So this plant is shown 30th March 25. See April 9 25. See more fruits are there. Fruits are ripened. This is April 21, this plant. Now the flourishment or the leaves are all still present, but they're very big leaves, and so these small weakness present. Even then the plant is very healthy, no leaves shedding, and also the fruits are also so much healthy. Now, what is the order I say? These are all medicines which I used and separately because these medicines, this is a test last plant. I used so many medicines in earlier years, and now this plant is a separate testing plant. First, I used berries, then alumina, then antium tart, then pulsatilla, then sepia, then meserium, then axomica, then mersol, and then phosphorus. These are all plants for these 2.5 years. This plant is still there. There is no shedding, but all these flowers, all these phosphorus, including short typical changes. That changes are all not to be not recorded at that time because it was also in an experimental way, experimental period. So all these plants now lost phosphorus, even the plants suffer or is adapted, and no change in this general but general healthy after the antim tart and pulsatilla. This antim tart and pulsatilla third medicine when applied, this plant becomes flourish and flourish. This flourishness. This after this flourishness, flower flowers appeared, this plant becomes more healthy and it has become this type. Even now, this plant is not healthy. Is healthy. That's very simply I am saying to see to hear to see to understand something about these medicinal applications. See, this is a butterfly. This plant, a root. See, the roots are there. Roots are there. See, within the cup, some water there. In Idiki district, when I was working in a very rural area, here there a plant, these roots appeared in some medicines, and I put a small butterfly cocoon. There the butterfly healthily flown out. It's simply, this is not something. Here, the next one is vitality of plants. What is vital force? Vital force and vital energy. The plants also have vital force. That is why the life is existing. Why the medicines acting or how the medicinal plants are absorbing and using these plants as nutrients or some other parts. Exactly, our homeopathic medicines do not have a nutrient effect. Exactly, they are doing the result is nutrient action. So many other actions are also, they are prohibiting the diseases, insects, by applying or producing some other things as natural plants. Here also I want to emphasize this enzymatic action, hormonal action, elementary. These three actions are even in a life, in plants also. These are all for survival, growth, reproduction, and photosynthesis, like many metabolic activities. These all because of these elemental substances, nutrients which absorbed from the roots and from the air and sun. So these are all materialistically, we see that these are all or the metabolism helping. But how these our dynamic medicines also have certain changes from to the plant leaves and shows their typical meaning crystal changes in cellular level. That is what I want to say. All regulatory stimuli, then gravity, symbiotic nutrition. What is gravity? These all life which are rising on Earth, living against gravity. Against that means they are coping with gravity. We are all. And the symbiotic, some other plants, near the plants, some other elements, everything, they are symbiotic. This is the symbiosis of the plants. The nutrition also. Nutrition we know, that is simply not the taking the food, but it is the assimilation and complete metabolism of a food is called nutrition. For all these happening because of the vitality of the plants. Then, what are the enzymes, especially so many enzymes, very speedily I will say amylase, phosphatase, lipase, this amylase, the phosphatase, lipase, phytase, phosphorus, cellulase, protease, urease. These are all also present in plants also. This phytase, especially phosphorus for absorption, they are secreting from the phytases from the root tips and to the soil for absorption of easy absorption of phosphorus. They are helping. Amylase, producing this is helping the digestion of these nitrates. Then phosphates, lipase for the fatty substances to digest. Cellulase for the cellulose and their metabolism. Protease for proteins. Urease for urea. These are the enzymes, like what to say, because of all are live animals and plants. See here, one chili plant I want to show. This plant's name is Phileas blue pepper. These blue peppers plants or blue chili plants are used my experiments because of easy to see the color and their changes in fruits and their abundance of flowers and fruits easily accessible. See the next one, the flowers. Here, I want to say another one. This is a chili leaf. You can see a leaf which is made into solution and absorbed. These crystals are common crystals. This is a crystal you can see, this is a crystal. This is a common crystal of a chili leaf. Everything in any chili plant, if you made a solution and see under microscope, you can see this type of crystal. But this is the one appearance. The next one is, see the same one. See, this is the same crystal. See, these angles, that is what I want to say. These same angles, these two crystals join. The next one, see, this is the original appearance. Most of the slides, when I see under microscope, we can see see the angle, this angle. Here a small notch, that is a small, the second one is behind this one. So these two crystals, that's a type of building. It's very common. That's what I want to say. This is a common natural crystal of actually the natural, original, that is natural. See, this is another appearance, the same crystal. This is same appearance. This is the other one. The other one is inside the crystal. That is the natural one. So one word, that is natural. This is a natural crystal. See, I want to show this is a very important one crystal that I got it from a plant which is very much diseased. This is my confirmation. Why? The plants, when diseased, they show certain type of abnormal crystals which are new. This is a new crystal. That's the new, and it is not normal, that is not natural. This is what I want to say. For these crystals, so many crystals in my over the later classes, if I get a chance, again I will show so many crystals. This is the one. It is very important one. That's why I selected this one. This is an important unnatural crystal, very new, and it is got from a diseased plant. The plants which are no more diseased leaf curling. These plants' leaf were curled, and it was a measly white appearance. That was the disease. See, it is in go. Then next picture, this picture emphasizes the plant peperomia, which was kept in globule mixed solution, and after that the cells studied under microscope. See the disease, so many texture of the stomata and the cell membrane were dissociated because of this globule. There was no particular change here observed. You can see, see the globule. See, this is a small stomata you can see. See, it's very closed, very closed, very closed, not opening. The cells are almost died, decayed, and necrotic. So many cells are there, but healthy cells are very few. See the green is very. See, this is a globule one, which which I want to say. Then next one, plants and products. So many products are separated from plants. They are the secretions or the excretions. Maybe secretions and the excretions, so their products are all for their protection. They are all plants protection alkaloids, flavonoids, oils, resins, and latex. This is essential oils. These flavonoids, alkaloids, resins, latex. These are all the products from plants. We know. Phytochemicals are the name generally applied to these all factors. These major ingredients, alkaloids, nitrogen-bearing compounds are alkaloids. Their action are many pharmacological general and particular action. Morphine, ephedrine. Morphine is a product from opium. Ephedrine is a ephedra plant which is from China, and it is a direct one. And also some chemicals which are synthetically also using. It's a life-saving, very important remedy for thousands and thousands of persons daily. Ephedrine. Strychnine also we know from Strychnus nexomica. Nicotine from tobacco. Quinine from cinchona. Cocaine from coca. Atropine from belladonna. Caffeine, coffee. Then codeine from again from opium, capsaicin. What is this capsaicin? From capsicum. Capsicum is being blasted from Vinca minor. Vinca minor is a plant we know. This is a very common plant in India and especially in Kerala. And this is the only very important plant from this incrustin and vinblastin are the elements are estimate. Yes. These are the important alkaloids which we know. Then phytochemicals are other one which is resins. What is resins? A solid, semi-solid or solid amorphous soluble organic solvents, antiseptic stimulants, oleo resins. There are two types of resins. Oleo gum resins. There part of gum also is there. Asafoetida and myrrh are the oleo gum resins. Then oleo resins, ginger, capsicum, and cannabis. This is a glycoresin. There is a glycoprotein. Glyco means glucose. Glycoportion. Glycoresin is jalapa is an example. Then balsam resins contain high proportions of benzoic cinnamic acid. Balsam provides samples. That is flavonoids and some other ingredients. Polyphenolic compounds acts as antioxidants, anti-inflammatory agents, as pigments. Chamomilla, ginkgo biloba, rutin are having these flavonoids. Saponins, compounds that form with water, can be steroidal synthetic hormones or terpenoids. Ginseng, ginseoside. We know ginseng is ginseoside is a saponin. Next, tannins. What is polyphenolic compounds are called tannins. Bitter taste. They have bitter taste and astringent taste. Wound healing capacity for diarrhea, applying bind proteins and contract tissues. It bind the tissues and contract tissues. For also it is important for many therapeutic complaints. Okay. Oak bark is an important of tannin. Then cardioglycosides, we know this digitalis heart digoxin is an important one. cardiac glycoside. This is that. From digitalis, digoxin is the important glycoside. Then volatile soils are the important one because of this, their aromatic smell, and for most of the oils of aromatic, Ayurvedic, and other essential are obtained from these plants, volatile oils. Next, I want to show another type of crystals, that is changing crystals. What are the changes of the? This is a fresh peperomia. A peperomia plant, these so many plants, later I will show. These plants, tincture leaves are taken from fresh and made into solution for 8 days in spirit, and kept it. Then the solution is decanted and kept for 2 days. And after that, one drop is placed on a slide and kept it for 16 days. 6 days sometimes. Some plants are only 8 or 6 days. 16 days is very apt for complete crystallizing the materials in a dry circumstances. So it's a peperomia. See, the crystals were very rare. See, this is the common one usually seen. No other crystals are seen. This is a small, big, but almost size is this common. This is the normal actual picture of a peperomia plant. You remember later on there are some changes others. See, this is what I want to say is a very characteristic change which occurred in a similar plant from Silesia vernum applied. Peperomia plant which is kept in a solution of 4 to 5 vernum Silesia number 10 pellet, sequester 100, and a new plant is placed there. After 3 or 4 days, some plants may be showing some other signs. Here, the plants is collected after 7 or 10 days. The leaves are collected and made into solution. And I saw this very typical crystal of Silesia vernum peperomia plant. This is also so many other types of crystals also there, but this was a very important one. And no peperomia of the same side, same slide. I was done the experiment several times. That not good, but Silesia especially shows this type of small vernum crystals. See the new one. Here, these plants crystal says because of under coming my category, that is new crystal. It's a new crystal. It's not a normal crystal of the peperomia, but it is because of the induced one, because of the Silesia one. That's what I want to say. The earlier crystal, this one, this crystal, this see the peperomia fresh one, and this is the important one for the Silesia one. Now, another one, we cannot even believe. See, Silesia 51, 50M. Second slide. This is not the first one. This is the second one. I used this Silesia 50 as for very little pictures. See the shape. It's very much elaborated. The next one, next picture. See. See, this is the picture of Silesia 50M. I got this because of that importance. I showed this here. This small crystal. See the angle, a small angle here. It is not a normal one. A normal picture in a crystal, very slightly, typically small one. The next one is another one. Only see the pictures. There's so many, Silesia 50M. Then next one is, this is a picture typically got from the plant which I experimented with a mixed SAR. Dr. Latif Sar's new medicine. I got it from the shop, and I used these pellets for experiment. I saw at that time, and I also sent this picture to him when he was alive. This mixed SAR. See the peperomia crystal change, little big. Here not new, but induced, but change, not null, not normal. Normal, but not abnormal. This is normal. Big size. The size become large because of the nutrients. See the other one. This is chili. This is a normal chili plant. I showed this crystal. This was a crystal. This crystal was there. See the earlier I showed this crystal. Likewise, there is some other additions, some small crystals also on that same crystal. That is why this shape is attained. This shape. You can see, I think that you can see these pictures. Hello? Hello?
Yes, sir. We can
Yes, sir. Okay,
see
okay.
everything is
Okay,
fine.
thank you. This one is also another one. Like here, you can see this is a plant Mixar peperomia in. This is an example of the big one and also abnormally normal. No, sorry, normally abnormal. This is a crystal. And characteristic normally, it is a normal crystal that is the stripe on one shape, everything. But what is the abnormality? See the clarity, fixity, then its contour, everything is shape, etc. Very clear. But the clarity is not the center, it is something very much degraded like. This is a crystal. Then biomix. What is the biomix? Biomix, certain biomix is a mineral mix bought from the this shop, vegetable shop, and after applying, there is so much, so much changes there. They are fertilized because they are fertilizers. Even then, they are the crystals were not having any particular change. This is my experimental plant peperomia, and I am using these plants and giving medicines and trying to say the essential element of given plant, peperomia, essential elements given. See, natural, one natural crystal. This is natural. It is natural, but no, not from this one. It is a natural of some other plant. That is, some other plant means it is a type of type kammal chedi. I think that this name a small fruit inside this very great good pea size, kadala size one. That's the name I got. I am now not remembering that one. That see, this is totally abnormal. That is a normal one, but it is not abnormal. Looks like abnormal, but it's natural. These crystals are not bounding like this. Only we will get this type of, because it's also a saponifying taste of that liquid. Everything that fruit, the small one, which is very small sized one. Inside that there is a very globular one. Inside that there is a small seeds are there, very sweety, and now it is very valuable fruit. In our elder days, our childhood days, it was in a cemetery, everything, and in our mosque areas, they will and we eat so many times. This is important because there will be not be a concise picture. This is the natural one. This is the normal one. There is no change. This is but totally abnormal because of this is a crystal obtained from Mixa. That is Dr. Latif Sar's crystal. Sugar is there. So many plants accepted. What are the things there? So many combinations. But even then, the crystal is completely changed abnormally. This got a very important one. This is abnormal. This also I sent to Mr. Dr. Sar when he was alive. This is the picture on the other side. This crystal is the view of other view. That is second view. This crystal, I think you can see. Can you see this? Yes. Next one. See, this is natural sugar of milk crystals. Yesterday also in Malayalam class I showed this is natural normal crystals, sugar of milk. This is biomix. What is biomix or mineral mix? The earlier one picture I showed, see the same one. The small crystal I want to show this. This is a chili plants applied biomix. This see, this crystal. This crystal is the natural normal crystal of chili. There is no particular change. That is what this biomix and availability of the nutrition and the compatibility of the what is called amicability, what compatibility of the systematic wholeness, fullness, like a nutritional level. There will be not be any change. The plant also will not have anything. They normally produce crystals, normal leaf structure, everything. This is normal. Biomix is a normal one, soothing for them, but homeo medicines are not soothing for them. They are actually, we are inducing something to them. That is what I want to say. See the one, this is salt. This is iodine salts, iodized salt. This is the crystals of iodized salt. It is never, it becomes, this is also iodized salt. This is also iodized salt. So this is something very not mimicking the sodium chloride, but it is having a little change. That is naturally not, but abnormally not. It is unnatural. It is not normal. This type of crystals are not normal. See, this is a normal crystal. We know this is sodium chloride, but it is iodized salt, some other crystals. So many things are there. In iodized salt, then another one I want to say this P green, small p. This is a p, very good sample for specimen for our experimental studies. See, very good, very little time, and may show so many results within time. This is only 7 years or 15 years. This is one. Then I want to show this tinospora. It's to see for your knowledge in this for the crystals, the number of crystals. That is the crystal of tinospora, normal one. Normal crystals. This also is normal crystals. This also normal crystals. This also a normal crystal of tinospora cordifloria. See, this is another normal crystal, tinospora. Last day in Malayalam class, I showed these plants and these crystals. Then peperomia mineral in. This is another one because the plant is produced inside the, this is put inside the mineral solution, and after that, this plant is weakened and observed under microscope. Mineral in same. See, a small crystal is there. This small crystal, there is a small change, but it is not an observable one. So many crystals are lacking. So new crystals may be there, but so many crystals are lacking in some cases. That is very important according to us, because we have to know what crystals are abnormally placing in the diseased area and what abnormal crystals are to be removed from that area. That only enough for the importance of our studies. So many things are there. Last I will say that this crystals are some other things. Okay. This one, this mineral makes in peperomia. What is mineral makes? Same, similar one. Mineral makes similar one. No particular change. See, no particular crystals are there. So many crystals are studied. After that, I selected this. So this is a bunch of small crystals. There is no particular change. That's what. This is one, the normal one. See, fresh in water. Fresh in water. What is it? This is a stomata studied fresh in water. This is fresh in water. You can see the stomata are full because to avoid water from these cells. So it is swollen. Word cells are very swollen, and the leaves are very greatly healthy, green but little whiteness because of the watering. This is also a peperomia plant, normal plant. Stomata of Silesia used to plant. That is the change. See the green preserved. That is the first one we have to notice. That Silesia preserves green chlorophyll. The stomata are half closed, not fully open. Water is completely, because for 3 or 4 days, 5 days, water may be completely dehydrated. Water, it is not dehydration. I am saying that Silesia do not dehydration. Actually, what is happening is the expelling of the water molecules is then doing, and that is why this echoing for complete voice absorbing areas also places this very big silica cubes or silica particles are using in the rooms for drying in some bottles with some other medicines. everything Silicea is used. Here the Silicea stomata, you can see the stomata changes. It's a very important one regarding our studies. This is another one, fresh in water. Again, see the swelling, not seen the stomatal opening. This is a Mysterium plant. Mysterium. See, this angle, I want to show a crystal which is typical one for the Mysterium. Second slide. This is also a Mysterium one. Same crystal, Mysterium second. Mysterium second means this is in one. See this angle, I want to show this angle. This angle shows in some another, some one plant, that is our that is what is called the puli. We have in Malayalam puli. We use this small tartaric acid crystal. This may be not a acid crystal, I know. But acids are showing this bar lake or crystal lake, and this sun lake radiated, very elongated crystals. This type of crystal will not be an acidic one, acid one, but it is likewise. This angle type of crystals are seen in that picture, that leaf of that substance. See, this is one very big stomata. Stomata, a normal peperomia in water. It is a normal peperomia in water, because in bottle. What is stomatal conductance? Dry air reduces water availability. Light increase the conductance of to facilitate photosynthesis. Then carbon dioxide, high carbon dioxide closes stomata. These are the changes are called stomatal conductance. In dry air, we know all these causing to reduce water dehydration. In potassium, have some control on leaves, edges curl, general chlorosis. See, this is the branch showing certain curling, the leaves, the dipping of the central areas of this leaf, and this punched out leaf. Half later we can see the half deco, that is a deadness. Half leaf, this half portion will be later on will be decay. That is also a importance of this kali potassium. This is kali more 30 used plant. See the change. This is a plant crystal, separately big crystal, because of kali more. See the same kali more 30, same kali more, an elongated crystal. This is also a same kali more 30. Then same solution. This is a clover solution. This is in extra introducing one for a clover, a plant clover. See the radiating acidity. I want to show by this a severe acid small leaves used for this gastric complaints, everything for in our natural medicine. See the crystals, one radiating here in all directions. This is an acidic crystal of clover. This is not succused 1 year clove solution. See the difference. This is the normal one, but not succused. The earlier one is succused and shown. This is calendula after 100 succussions, a tincture. I have shown the picture of calendula in earlier classes, very great, opaque, very long, elongated, like this clean one, like area, elongated. But this is after succussion, what happened? This is an important one, this is tincture. Then the spirit, 28 to 124 to 1 to 24 in solution. Then 9124 solution and decanted. Then 15.5, 24, 25, the study of this picture taken. This is spirit, only spirit. This is a deviation. You may say that spirits may change the plants. Yes, sometimes. Only the spirit can, plants only cannot survive because it's a very important one. According to plants, this methyl alcohol and ethyl alcohols are very important chemical substances same as to their decayed products. So these plants may respond to spirit also. So the study of homeopathic medicines in drops is a very important discovery. Then fresh land peperomia. This crystal, a triangular crystal of this land peperomia. This is an abnormally, this is natural one, but naturally abnormal. From land, sometimes some other supportive medicine or some other symbiotic effect of some other nearest plants. This crystal of peperomia. This is also the claw, important one. This is not normal. Land fresh, not normal changes. Land from land, we can see some other crystals also sometimes. And fresh land here also some another one. Then anti-crude chili and anti-crude chili become given anti-crude strong and it is absorbed. This crystal. See, no changes. The anti-crude is a very important element which consumes plants in a large amount. Or sometimes or it not absorbs. But from the presence of this very small amount of anti-30, not anti-crude, antimony is enough. But especially a very good potency is anti-30 will definitely give, if the plant is in time for fertilization, it will flourish. Why? Antimony. No crystals are settled, but the plants very much gritty and the seeds, the fruits are not fallen off, fruits tight, calyx, and they are grit. These are everything. Not for especially flowering, but not falling off effect of this antimony. Fresh second land, then antimony. This is orilla. It is a separated pressure coming inside by way. Orilla. This plant's leaf always shows not orilla, but ori flower. A single flower-like crystal is definitely we see in orilla. And the background of this one, you can see a color, slight yellow. That is an important decoction. That is orilla. Orilla is a component of almost all Ayurvedic preparations. It's a very important healthy toning. And these background portions is very important. And that's why I used it. So many background solutions are typically showing their existence, their evidential performance. Because of that, we can have these plants become very much identical. Identifying these plants are helpful. In pharmacy, it is very important. This study. See, orina. Then other manjal, curcumin. This curcumin. These are some type of soluble crystals from curcumin. Base is there. This is curcumin. This is another one. Last day, so I showed this plant from another site. This is one. Then young leaves. Young leaves show certain typical changes. See the original picture, original crystal in this one. Young leaf. Old leaf. This is honey. I want to show. Last day also I showed this. You want have to say, see the octa 1, 2, 3, 4, 5, 6. It's like the honey bees planning. Honey. This is a clear honey picture. Honey. Today my class was so over. Very speedily I covered. So many changes I did in this, but now everything is disappeared. So many changes. Here also so many changes. And not see that. Oh, okay. The end. My class is now at the end. Today I confirmed within 10 minutes. Any doubts? Anything can ask. or I will say something about the earlier ones. This leaf, last day also I showed this leaf. I was so much interested in it. This is a big, small leaf, not our jackfruit, that type of one big tree from a hilly area. This leaf. I want to emphasize these veins. See, big veins. Studying this is an important one for crystals. There may be something which give some clues about these veins. Definitely it will show. And what is statoliths? Here are two things, maybe having some doubts about these crystals. These are the otoliths in human ear calcium. We know. What is that thing this calcium usually shows? These otoliths for our hearing, these otoliths, these calcium elements inside the cochlea will help to balance also. Likewise, statoliths are hearing root caps. They are not bearing. This is root caps bearing. What is the root caps? Root caps of all plants. Some, not all plants. These statoliths, there's calcium bound elements to absorb, to make a surrounding area of that plant to absorb. This sort of this characteristic uh, identity is keeping by berberis plant. It says that berberis plant produces something around its plants and it destroys and it does not allow the other surrounding plants to grow. So the roots and around the soil may have some action that is not berberin, but some other things, alkaloids, certain other alkaloids. So this is a crystal of this plant. It's near Munnar, Idiki, in a cold area. So many, two types. One is yellow flower, one is red flower. The plant is green, like this. I got some certain typical crystals of this leaf, resembling this leaf. That is another one. So this resemblance, what I want to say. Leaves, plants, everything shows these crystals. The small one. This is curcumin. This potassium gives turgidity to the leaves. That's why this, see, Kalimur applied plants, but it is so much increased, 6, not 30. Higher potency sometimes may give higher intensity, higher concentration. That is one of my observations. We can't assess this. We cannot accept this very easily. In biochemic textbook, one textbook that Smith, I think that the doctor's name is Smith or some other name, no one to remember. The doctor says, hello, now the
Yes.
video is, okay. That Smith, the Silesia is obtained from the wounds of some Silesia plants, Silesia ulcers. That's a very important. That is the basis of all these my studies. Hope we can confirm. Try. This is fresh land. Here also, a small. Everything is okay. Anybody can ask any doubts? Okay, I am completing this here.
Doctor, we can conclude the session and we can have second part later. We can have some 5 minutes discussion.
Okay.
Okay.
Thank you.
It's yes. It was really an excellent session. Unfortunately, the participants are comparatively less. I'm inviting Dr. Rambarasa sir to say a few words on this presentation.
Thank you, Dr. Dhaneji, and congratulations, Dr. Mashood sir. Very nice,
Thank you, sir.
excellent session. Agro-homeopathy. Agro-homeopathy or homeopathy plant also possess a dynamic life force. Dynamic life force. Plant is also possess dynamic life force. And growth, healing, response of environmental. So, homeopathic for plants is based on the same principle used in human homeopathy. Similia, vital force, and potentization, minimum dose, individualization, same process. And individualization, according to homeopathy, in human mental symptom, physical symptom, the plant individualization, soil condition, and type of stress, health, and a stage of disease, plant reaction, etc. So, lot of medicine, yani shock, plant shock, arnica and calendula, fungal disease, sulfur silicea, silica, and big growth, calcarea, fos is the best remedy according to the symptom growth. And thought, stress, datramil, just like human treatment plant agropathy. So, very, very thanks, Dr. Mashood sir. Thank you.
Thank you.
Now I am inviting Dr. Bob Lim to say a few words and the vote of thanks. Dr. Bob, please unmute yourself. Yes. There was some problem. Okay. Yes, Dr. Bob, are you there?
Sorry.
Yes. You
Sorry,
can
I have to close the other webinar. I'm under two webinar. I'm very, very sorry. Very busy today. I just
Okay.
went over to East Malaysia, Sarawak. Okay. On behalf of the IPH, I would like to say a big thank you to Dr. Mashood for his research in this field. Is very, very highly technical. I think he's one of the only one in the world doing this type of research. Then I hope that the scientific community in the global world will recognize his research and also at the same time give a greater promotion for homeopathy to be recognized as a scientific school of medicine. So, on behalf of IPH, I would like to thank Dr. Mashood for this tonight's highly technical webinar. And I hope that he will present more of his research and sharing with us in the near future. Thank you very much once again. I hand it over to Dr. Danesh.
Okay, thank you, Dr. Bob Lim, Dr. Mashood, and those who attended the session. And tomorrow we will have a session from Dr. Vansh Lunia, MD, and he will be talking about when smaller remedies come to rescue in surgical cases. That means we can have a study on rare remedies from Dr. Vansh Lunia. Thank you all. Good night. It's over to
Thank you
Dr.
all.
P. Shaila to moderate the Malayalam session.